On behalf of the Pancreatlas team, I'd like to thank the organizers for allowing us to present and to all of you who have logged on to listen. My name is Diane Saunders, and I'm excited to tell you more about Pancreatlas and its open source component. Just some brief biological introduction. The pancreas is a bifunctional organ that's part of both the digestive and endocrine systems. In the past decade, many research programs have been initiated to identify causes and treatments for diabetes, pancreatic cancer, and other conditions. Among the many varied data types generated by these initiatives is cross-sectional imaging data that aims to better understand tissue architecture and the spatial relationships between cells. What this means in practice is analyzing hundreds of tissue blocks in various formats, generating thousands of images in different modalities. A few years ago, our group began thinking about how to compile all of this data in a meaningful way that could link the de-identified clinical information with high-resolution images that could be easily accessed by the community. So some of the challenges we faced obviously included the image quantity and size, meaning we wanted to develop a platform that allowed people to easily access the images they were interested in. We knew we would have a lot of different varied file types, and also that we wanted to annotate these images with pertinent metadata. Finally, we wanted biologists to be able to actually interact with these images rather than just displaying them in static figure panels in publications. To address these challenges, we assembled a multidisciplinary team that includes biologists, developers, and bioinformaticians. And having these groups in the same room from the very beginning has, in our opinion, really been instrumental to the platform's success. So the goal was to build a tool that was easily accessible to the biologists, but also could be customized and utilized by analysts, developers, and data scientists. It was important to us that we not reinvent the wheel, and that we have a user-friendly and online tool that wouldn't require any additional downloads or installations. We also wanted to make sure that our platform was as scalable and flexible as possible. We began with the backend infrastructure of Omero Plus, which is the licensed version of OME, the platform that takes advantage of bioformats that many of you may be familiar with. This was connected to our institutional infrastructure, as well as our laboratory information management system and dedicated image storage. We then built a custom web interface, which seamlessly points users to Path Viewer, which is the image viewer associated with Omero Plus and similar to the OME iViewer. What this allowed was that our users could interact and access these fully featured components of the Omero system, but also have easy curated points of entry to start exploring. If any of you have used the OME web browser, you know there's a bit of a learning curve to access relevant image and study metadata. So our goal was to leverage the platform's features and put them in the hands of a more casual user with as little friction as possible. So to do this, we created two kind of different components. The first was an API, and the second was a user interface. So this React-based user interface was geared towards biologists. And to connect to Omero, we built an API that could handle metadata retrieval and could also interface with different data sources. If you're listening, I'm guessing that you are involved in either image generation, analysis, or image sharing, or maybe all three. So here's how we hope that our group can help. The framework that we built for Pancreatlas is an open source tool, and it's available on GitHub. You can scan the QR code here or visit this link. And we named this platform FFIND, or Flexible Framework for Integrating and Navigating Data. So we really believe that it's a data agnostic platform, and it can be used for not just imaging data, but also other types of data. And the video that you see playing here illustrates that it really only takes a couple of minutes to get this up and running, and there are even some mock data sets for you to play with. One of the key features of FFIND is that it has a spreadsheet-based metadata import system, which is friendly to most biologists. So these columns in the spreadsheet represent researcher-defined variables, and then each row of metadata corresponds to one image. And what happens when these images and the metadata are imported is that the columns that you had previously defined actually quickly become filters, whereby the users can drill down to the information that they're looking for. And in Pancreatlas right now, our current collections feature over 2,000 images, and some of these are geared and organized around manuscripts, while others are coming from phenotyping programs of various different efforts. And now I just want to show you some of the FFIND features in action on Pancreatlas. To give you a quick tour of the website, pancreatlas.org, from the homepage, you can either click image collections or the down arrow to view all datasets currently available. If you'd like more context, select collection details to read more about the project, or you can simply choose a collection and begin browsing. Once in a collection, you'll see a grid of available images, each of which has its own card that lists important donor and sample characteristics as well as the different markers visualized. On the left here, you see metadata filters, which will refactor automatically to give you available images as you select different attributes. Clicking on the magnifying glass launches a modal preview, which enlarges the image thumbnail, gives some additional sample information, and then allows you to navigate directly into Path Viewer, which is Glencoe's web client for exploring images. So this particular image was acquired using Codex technology, which is a fluorescent immunohistochemistry-based multiplex system. This is a cross-section from a non-diabetic adult donor, which is full resolution at 20x magnification and contains 28 registered channels. So notice that the image is loading almost instantly despite being more than 25 gigabytes, meaning that you can immediately browse and interact with multiple images within a given data set without the friction of long download times. And instead, Path Viewer really lets you dive straight into the data. So first, I want to just briefly highlight the list of markers to the left. These are grouped into biologically relevant categories. So not only can I pan around the image and explore areas of interest, I can also toggle on and off channels depending on the cell type that I'm interested in. So for example, right now the endocrine cells, hormone-producing cells are shown, but if I wanted to, I could add in a vascular marker to visualize islet capillaries, or I could choose an immune marker in order to understand the proximity of macrophages to various structures within the pancreas. And from the header menu, you can easily access our nomenclature tab, where we've provided accessible descriptions of terminology and have tried to really standardize metadata across our collections. Hopefully that gave you a little taste, and you'll go explore pancreatlas.org further. And I wanted to just briefly end by talking about some of the features that we're excited to add in the future. So one of the things that is in the works right now is that in addition to curated collections, there will soon be an option to view all data sets together. And so that will bring all of the imaging data into one grid and allow users to search and filter across different images and collections based on what they're interested in. And also we're working to improve the layout of some of these specific filters. And all of this development work will be added to the FFIND GitHub repository. And I also mentioned that FFIND has stubs in place for non-imaging data. And so you can think about this as kind of a platform that will enable you to share multiple different data sets in whatever data viewers you like. So for example, in the pancreas, we often do imaging as well as isolating individual cells and doing sequencing. And so what you see right now in Pancreatlas is that the cards kind of displayed in a grid view preview the images, but you could also envision these cards displaying the preview of a sequencing data set. And so clicking on those cards would launch into a different viewer rather than launching Path Viewer. But the value here is really having this one platform that houses all of the data in the same kind of format and annotations so that people can easily view and access data sets that they need from one place. So I want to thank, obviously, the entire Pancreatlas team. This has been a really fun project to work on, and also our collaborators at Glencoe Software. You can see that there are a lot of people involved in an effort like this, but we're also a small team. And so one of the reasons we like FFIND is that you can easily implement it even if you don't have a dedicated team just working on an image database. We'd love to hear your feedback. We look forward to more conversations after the talk and during the conference. And please don't hesitate to get in touch if you have questions. Thanks so much.